Hello everyone and welcome to our channel, MooTechie. It's wonderful to have you here. Today, we're diving into the fascinating world of proteins, exploring their structure, types, and functions. We'll learn with clean visuals and animations, so be sure to stay till the end for a quick summary. Now, let's begin our journey into proteins. Structure, types, and functions. So, what are proteins? They are fundamental molecules of life, driving biological processes. We'll see how they're built, from amino acids to complex quaternary structures. We'll look at their classification and types, like simple, conjugated, and derived proteins, and the difference between fibrous and globular forms. And finally, we'll uncover their vast biological functions, from acting as enzymes and hormones to transport and structural roles. Now, let's move on to the fundamental question. What are proteins? Proteins are polymers of amino acids linked by peptide bonds. They fold into specific functional three-dimensional shapes. The principle of structure leads to function governs their activity. They are ubiquitous, found in enzymes, antibodies, muscles, and tissues. Now, looking at this diagram, we see a detailed view of a protein structure. We have an entity showing a DNA double chain and mixing. Next, we explore the very building blocks of proteins. Amino acids, building blocks. They have a common core, an amino group, a carboxyl group, a hydrogen atom, and a unique R group. There are 20 standard types classified as essential versus non-essential. It's the R group that determines the amino acid's properties, like whether it's polar, nonpolar, or charged. And importantly, proteins are synthesized in a biological direction, from the N-terminus to the C-terminus. So how do these building blocks connect? Peptide bond formation. This is the condensation reaction that links amino acids. It's a condensation reaction linking amino acids by releasing a water molecule. This is also known as dehydration synthesis. This process forms the polypeptide backbone, which has a repeating pattern of nitrogen, alpha carbon, and carbonyl groups. The peptide bond itself is planar and rigid due to its partial double bond character. And remember, proteins are synthesized directionally from the N-terminus to the C-terminus. The diagram here illustrates this. We start with an amino group and a carboxyl group from two separate amino acids. Through a dehydration synthesis or condensation reaction, a peptide bond is formed, creating a dipeptide and releasing a water molecule. With the chain formed, we can now look at its levels of organization, starting with the most basic. Primary structure. This is the linear sequence of amino acids, which is genetically determined. This specific order determines the higher level folding and the final shape of the protein. It is written and read from the N-terminus to the C-terminus. A single change in this sequence, like in sickle cell anemia, can dramatically alter the protein's function. The image shows several chains labeled A, B, C, and D, representing different polypeptide sequences. Now, let's understand how this linear chain starts to fold. Secondary structure. This refers to the local folding patterns within the polypeptide chain. One common pattern is the alpha helix, a coiled structure stabilized by intramolecular hydrogen bonds between the backbone carbonyl and amide groups. Another is the beta-pleated sheet, where strands lie side by side, connected by hydrogen bonds. These sheets can be parallel or anti-parallel. The driving force behind these structures is the hydrogen bond formed between the NH and C equals O groups of the peptide backbone. We also have turns and loops, which are flexible regions connecting helices and sheets. The amino acid proline often disrupts helices, introducing kinks. Following this local folding, the protein adopts its overall three-dimensional shape. Tertiary structure, 3D folding. This is driven largely by the hydrophobic effect, where nonpolar groups bury themselves inside the protein while polar groups face the watery environment. The structure is stabilized by disulfide bridges, ionic bonds, and hydrogen bonds. Proteins often have modular domains or motifs that act as functional structural units. It is this final three-dimensional shape that determines the protein's specific biological function. Now, some proteins are even more complex, involving multiple chains. Quaternary structure. This is the assembly of multiple protein subunits into one functional complex. It's stabilized by non-covalent interactions and sometimes disulfide bonds. This level of structure enables cooperativity and allosteric regulation, where a change in one subunit affects the others. A classic example is hemoglobin, which is a tetramer with 4 chains. The diagram here shows a quaternary structure with various subunits. We can see alpha helices and beta-pleated sheets within the individual chains, and the entire complex is held together by various interactions like hydrogen bonds between different parts of the structure. Now that we understand their structure, let's see how we classify them. Protein classification. We have simple proteins, which are composed only of amino acid residues like albumin and keratin. Then there are conjugated proteins, which are combined with non-protein prosthetic groups. Examples include hemoglobin and lipoproteins. And finally, derived proteins, which are products of hydrolysis or modification, like peptones and coagulated proteins. We can also classify proteins based on their overall shape and function. Fibrous versus globular. Fibrous proteins are structural and stable. They have an elongated strand-like shape. They provide structural support and protection, are generally insoluble in water, and examples include collagen, keratin, and silk. On the other hand, globular proteins are functional and dynamic. They have a compact spherical three-dimensional shape. They perform metabolic functions, are mostly soluble in water, and examples include enzymes, hemoglobin, and insulin. But what happens when a protein loses its carefully crafted structure? Protein denaturation. This is the unfolding of protein structure leading to a loss of function. It involves the disruption of secondary, tertiary, and quaternary levels, while the primary sequence remains intact. Physical causes include exposure to extreme heat or UV radiation, which vibrate atoms and break weak bonds. Chemical agents like changes in pH, high salt concentrations, or organic solvents can also disrupt these crucial interactions. The functional impact is a loss of biological activity. Enzymes lose their catalytic ability, and this process is irreversible in many cases. The graph on this slide shows SDS-induced denaturation of a protein called GFP at pH 6.5. The y-axis shows intensity, and the x-axis shows time in seconds. We can see that as time progresses from 0 to about 175 seconds, the intensity drops significantly, indicating the protein is unfolding and losing its native structure. After all this, let's remember why proteins are so essential to life. Biological functions. They are crucial for catalysis. Enzymes like amylase speed up metabolic reactions. They handle transport. Hemoglobin carries oxygen, and ferritin stores iron. They are key in signaling. Hormones like insulin and receptors transmit data. And they provide defense and structure, like antibodies and the proteins actin, collagen, and keratin. Now, let's do a quick recap of everything we've learned. Summary key takeaways. First, the building blocks. Amino acids link via peptide bonds to form polypeptide chains. The structure hierarchy shows that function is determined by 4 levels, from primary to quaternary. Proteins are categorized by composition, like simple or conjugated, and by shape, like fibrous or globular. And finally, stability and function are linked, as denaturation causes loss of structure and function. Thank you so much for watching. Did this video help you understand proteins? Don't forget to support the channel by liking, subscribing, and sharing. We'd love to hear from you, so please comment below and let us know what topic we should cover next. Thank you. See you in the next class.